I want you to picture something for a second.
Yeah.
Imagine you're standing in a neonatal intensive care unit.
Okay.
And you're looking at a newborn baby. And just observing from the side of the crib, this infant is doing everything a newborn is fundamentally supposed to do.
Right. The basics.
Yeah, exactly. They smile. When they get hungry, they cry. You offer them a bottle, and they immediately root and suckle. You watch them move their eyes to follow a light, make different facial expressions, kick their little arms and legs. To anyone watching, this is a beautifully expressive, active, totally typical baby.
Which honestly makes the reality of the medical chart on the end of that crib almost impossible to comprehend.
It really does.
Yeah.
Because imagine looking at that chart and learning that this infant was actually born entirely without a forebrain.
Yeah.
It's this tragic, incredibly rare condition known as hydranencephaly. And when you dive into the medical literature surrounding these cases, it creates a profound cognitive dissonance.
Oh, absolutely.
Because we are taught, culturally and scientifically, that the forebrain, that massive folded cerebral cortex, is the absolute seat of who we are. We're told it's the engine of our higher cognition, our behaviors, our humanity.
Right.
So if that massive structure is missing entirely, what on earth is orchestrating all of those incredibly complex, deeply human behaviors you're watching that baby perform?
It's basically the ultimate biological paradox. And the answer really challenges everything we think we know about neuroscience, because the orchestrator of all those behaviors is the brainstem.
The brainstem.
Yes. And what we're going to realize today is how catastrophically we have underestimated this structure. I mean, in popular culture, and honestly, even in a lot of introductory biology classes, the brainstem gets dismissed with this almost condescending term,
The lizard brain.
Right, the lizard brain. Like, it's just this ancient, unthinking machine. Exactly. It's treated as this primitive, archaic stock of tissue whose only job is to just keep your heart beating and your lungs inflating, while the glamorous, highly evolved cerebral cortex does all the heavy lifting.
Well, today we are dismantling that myth entirely. We are starting a broader journey into the highest levels of human behavior. It's a series exploring learning, memory, language, and cognition, and we're pulling directly from Part 6 of Principles of Neuroscience.
It's a fantastic textbook.
It really is. But to understand the top floor of the mind, we have to start in the basement. So we are taking an advanced neuroscience masterclass on Chapter 40, which focuses exclusively on the brainstem.
And it's a dense chapter.
Oh, very dense. Our mission today is to completely redefine how you think about the base of your skull. We're going to map out how it interfaces with the physical world, look at the astonishing way it builds itself in the embryo, explore the hardwired software programs that control our survival, and finally, uncover the hidden chemical systems it uses to modulate our entire state of consciousness.
There is a lot to cover.
There is. So let's unpack this. We have this idea of the brainstem as a supercomputer. And to understand a computer, you have to look at the cables plugging into the back of it.
Right, the wiring diagram. And to appreciate the brainstem's wiring, we have to contrast it with the spinal cord. Because the spinal cord is a brilliant piece of engineering, but it essentially taps out at the neck.
It just stops.
Yeah, it only reaches as high as the first cervical vertebra, basically C1. So it handles the sensory and motor innervation for the limbs and the trunk. But from the neck up, the spinal nerves are done. The cranial nerves have to take over completely.
And when we say cranial nerves, I think the immediate assumption is that they just handle the cranium. The skull, the face, the eyes.
Sure, that's what the name implies.
But the deeper you look at this anatomical map, you realize the brainstem actually bypasses the spinal cord entirely to reach deep into the body's internal cavities.
That is a crucial realization. While the cranial nerves absolutely provide somatic and visceral innervation for the headman, handling conscious sensations, muscle movements, and internal state monitoring to specific nerves completely break that geographical boundary.
Which ones?
The glossopharyngeal nerve, known as cranial nerve IX, and the vagus nerve, which is cranial nerve X, they extend their control far beyond the neck.
Wow.
Yeah, they project deep into the chest and supply visceral, sensory, and motor innervation to the heart, the lungs, and most of the abdominal organs. Essentially, they manage everything down to the pelvic region.
So the brainstem has a direct, high-speed fiber optic cable running straight into the gut.
Exactly.
Let's walk through how these cables actually originate, because the architecture is highly specific. The textbook lays them out in rostrocaudal sequence.
Right, meaning running from the front or top of the brain down toward the tail or bottom.
Right. And there are 12 cranial nerves in total, numbered 1 through 12. But the very first two seem to operate on a completely different set of rules from the rest.
They do. Cranial nerve A is the olfactory nerve, which handles our sense of smell, and cranial nerve 2 is the optic nerve, responsible for vision. Unlike all the others, these two bypass the brainstem entirely.
They just skip it.
Completely. They act more like direct extensions of the brain itself. The olfactory nerve enters directly at the base of the forebrain, and the optic nerve routes into the diencephalon. However, the remaining 10 cranial nerves, so 3 through 12, arise directly from the brainstem at highly characteristic, very predictable locations.
And when you look at a structural model of the brainstem, there is a bizarre anatomical quirk that immediately stands out. Almost every single one of these brainstem cranial nerves exits from the ventral surface.
The front side of the brainstem, yes.
It's an incredibly uniform design,
How about
except for one road nerve.
yes. The trochlear nerve. Cranial nerve, the fifth. It is the lone exception to the ventral exit rule.
What does it do?
Well, instead of emerging from the front, it sneaks out of the midbrain from the dorsal surface, the backside, just behind a small structural bump called the inferior colliculus.
Okay, so it comes out the back.
Right, and once it exits the back, it has to physically wrap all the way around the lateral surface of the brainstem just to join the other nerves heading toward the eye.
It's like a contractor wiring a house and deciding to run one single cable out through the chimney instead of the basement, just to wrap it back around the porch. I mean, is there a functional reason for this, or is it just an evolutionary leftover?
It's largely considered an artifact of evolutionary development, honestly. The brainstem folds and thickens so dramatically during embryogenesis that some primitive pathways end up taking what looks like the scenic route in the adult brain.
That's so strange.
It is, but despite the strange route, its function is hyperspecific. And that brings up how precisely the brainstem delegates tasks. Ocular control is a perfect example of this. Moving your eye isn't controlled by one master nerve. The labor is heavily segmented.
Break that division of labor down for us, because it's not just one wire.
No, not at all. You have 3 distinct motor nerves collaborating to aim your eye. The abducens nerve, cranial nerve valsalaic, has the most straightforward job. It innervates a single muscle, the lateral rectus, which basically pulls the eye horizontally outward, away from your nose.
Okay, pretty simple.
Then you have the rebel we just discussed, the trochlear nerve. It also innervates just one muscle, the superior oblique, which depresses the eye and rotates it slightly inward.
So those two are highly specialized.
Exactly. And finally, the arculomotor nerve, cranial nerve deli, does the heavy lifting. It supplies the remaining 4 extraocular muscles, it physically lifts your upper eyelid, and it carries the parasympathetic fibers that actively constrict your pupil when you're in bright light.
So you have this delicate triad of nerves working perfectly in sync just to let you glance across a room.
Right.
But then on the completely opposite end of the spectrum, you have something like the trigeminal nerve. Cranial nerve V. I mean, this isn't a delicate single muscle wire. This thing is an absolute industrial workhorse.
Oh, the trigeminal is a massive mixed nerve, meaning it carries both motor commands out and sensory data in. It leaves the lateral pons in 2 distinct routes.
What's the first route?
The motor root is relatively small, and it handles the muscles of mastication. So every time you clench your jaw, chew a piece of food, or grind your teeth, you're using the masseter and temporalis muscles, which are driven by the specific root.
Okay, and the second route?
The sensory root is what makes the trigeminal so spectacular. It arises from an enormous cluster of neurons called the trigeminal ganglion, which is sitting right at the floor of the skull.
And from that ganglion, it splits into 3 giant branches, right? The ophthalmic, the maxillary, and the mandibular divisions.
Yes, and those 3 divisions collectively blanket the human face. I mean, they innervate the orbit of the eye, the nasal cavity, the forehead, and the scalp, all the way back to the crown of your head.
That's a huge surface area.
It is. And furthermore, they supply the entire oral cavity, the teeth, the gums, the roof of the mouth, the anterior tongue. It basically gathers pain, temperature, and touch data from virtually the entire surface of the face and the inside of the head, and it funnels it all back into the brainstem.
Now, earlier we mentioned the vagus nerve, cranial nerve X, and how it reaches deep into the abdomen. And when you look at the research on how the vagus nerve interacts with our digestive system, It completely reframes how we think about hunger and fullness.
It really does.
Because the vagus nerve isn't just an output wire telling the stomach to churn, right? It's actively listening to the chemical environment of the gut.
Yes, it is the ultimate biological feedback loop. The vagal sensory neurons physically innervate the lining of the gastrointestinal tract. They're constantly monitoring postprandial functions, the state of your body after you consume food.
Right.
And one of the most fascinating mechanisms detailed in the literature is how these vagal afferents regulate satiety, the feeling of being full, through a specific peptide called cholecystokinin, or CCK.
So when I sit down and eat a heavy meal, my stomach physically stretches.
Yeah.
But it's not just the mechanical stretch that makes me feel full, right? It's an actual chemical signal.
Correct. When fats and proteins from your meal enter the duodenum, which is the very first section of your small intestine, specialized enteroendocrine cells lining the gut wall detect those nutrients. And in response, they secrete the peptide CCK.
Okay.
But this CCK doesn't just float randomly in the blood. It specifically binds to receptors on the endings of the vagal sensory nerves located right there in the gut tissue.
And once it binds, it fires a signal straight up the vagus nerve, ascending through the chest into the brainstem, essentially yelling, we have enough calories, stop eating.
Exactly. It stimulates a profound systemic feeling of satiety. And this precise neurological pathway has massive implications for modern medicine, particularly in the realm of weight loss and bariatric surgery.
Really? How does it tie into surgery?
Well, bariatric surgery is currently one of the most effective long-term treatments for severe obesity. Historically, we thought it worked purely through mechanical restriction. You make the stomach smaller, so the person can't physically fit as much food inside.
Right, that's a standard assumption.
But the neuroscience suggests it's actually a rewiring of this exact vagal communication. The structural alterations made during a gastric bypass or a sleeve gastrectomy fundamentally change the way food interacts with the gut lining.
Which changes the chemicals.
Yes. It alters the secretion profiles of gut peptides like CCK. The current working hypothesis is that bariatric surgery recalibrates the responsiveness of these vagal efferents. So the brainstem starts receiving an amplified, much earlier I'm full signal.
Wow.
The surgery changes the anatomy of the stomach, sure, but the sustained behavioral change, the lack of hunger, is being driven by this reprogrammed vagal signaling to the brainstem.
Wait, so an incredibly invasive gastrointestinal surgery is actually, functionally, a neurological intervention?
Essentially, yes.
We're altering the gut to hack the brainstem's feeding software. That is a stunning paradigm shift. Before we move into the internal architecture of the brainstem, I have to bring up a nerve that almost feels like an anatomical practical joke. Cranial nerve AAI.
The spinal accessory nerve. It's definitely one of the strangest structures in neuroanatomy.
Why is it considered an imposter? I mean, it has a cranial nerve number. It sits right there on the chart.
True, but functionally and anatomically, it is a spinal nerve masquerading as a cranial nerve. True, cranial nerves originate from nuclei within the brainstem itself, but the spinal accessory nerve originates from a column of motor neurons located in the upper segments of the cervical spinal cord, way down in the neck.
So the cell bodies are in the spine.
Yeah.
By all logic, the axons should just exit the spine horizontally and go to the neck muscles.
Logic would dictate that, yes. Instead, these rootlets emerge from the side of the spinal cord, bundle together, and travel upward. They physically enter the skull through the foramen magnum, the giant hole at the base of the skull, where the spinal cord meets the brainstem.
So they go up into the brain.
They literally sneak into the cranial vault, only to immediately turn around and exit the skull back downward through the jugular foramen, traveling alongside the glossopharyngeal and vagus nerves.
Wait, just to go right back to where they started.
Basically, once it's finally back out in the neck, it innervates the trapezius and sternocleidomastoid muscles. Those are the exact muscles you use to shrug your shoulders and turn your head.
So puts on a fake mustache, sneaks into the highly exclusive cranial nerve club through the back door, grabs a drink, and immediately leaves through the front door just to go shrug his shoulder.
That is a very colorful way to describe it, but anatomically accurate.
Biology is just astonishingly weird sometimes. But it really highlights the complexity we're dealing with. We now know what these cables do, and we know where they travel in the body. But to understand how this supercomputer processes all that data, we have to open the chassis. We have to look at how these nerves are organized inside the brainstem.
Right, the internal architecture.
And to do that, the book says we have to rewind the clock all the way back to when we were just embryos.
Yes, and this is where the underlying logic of the brainstem finally reveals itself. Because if you just look at a slice of an adult brainstem, it looks like a chaotic jumble of nuclei and fiber tracts. It's a mess.
Just a pile of gray and white matter.
Exactly. But if you connect this to the bigger picture of embryonic development, you find a pristine, elegant blueprint. The nuclei of the brainstem, meaning the specific clusters of cell bodies where these cranial nerve either originate or terminate, they're organized in highly structured longitudinal columns.
Okay.
And this columnar pattern is directly homologous to the basic sensory and motor layout of the spinal cord. They share the exact same structural origin.
So let's visualize that origin. In a developing embryo, long before there is a brain or a spine, you just have this basic hollow tube, the neural tube.
Picture the caudal end of that embryonic neural tube. It is essentially a hollow cylinder. And running longitudinally down the inside walls of this tube, right along the central canal, is a distinct groove called the sulcus limitans.
The sulcus limitans.
Yes. This groove is not just a landmark. It is a fundamental developmental border. It divides the walls of the neural tube into two highly specialized zones. Dorsally, toward the back of the embryo, you have the LR plate.
And the LR plate handles what?
The LR plate is strictly dedicated to sensory processing. Ventrally, toward the front of the embryo, you have the basal plate. And the basal plate is strictly dedicated to motor output.
So the golden rule of the neural tube is sensory in the back, motor in the front.
Exactly.
And the spinal cord essentially keeps this basic tube shape into adulthood, right? The sensory dorsal horns are in the back, the motor ventral horns are in the front.
Yes, the spinal cord is very faithful to that original tube design.
But the brainstem doesn't stay a simple tube. It goes through a radical physical transformation. The central canal opens up.
It does. As the neural tube develops into the region of the medulla and the pons, the tiny central canal dramatically widens and opens up to form a large, diamond-shaped cavity known as the fourth ventricle. And as this ventricle expands, the lateral walls of the neural tube are literally forced to splay outward.
The analogy I keep coming back to is a massive server room. Imagine the neural tube as a long server rack, and you decide to physically pry it open from the back like a book.
That's a great visual.
Because the tube opens at the back, all the sensory equipment, the LR plate, gets pushed laterally out to the side walls of the room. But the motor equipment, the basal plate, which was already in the front, remains relatively medial, close to the center aisle.
That is a phenomenal way to visualize it. Function dictates the physical slot. Because of this splaying, the adult brainstem isn't organized front to back like the spinal cord. It is organized medial to lateral.
Which is side to outside.
Right. This splaying creates 6 distinct longitudinal columns running up and down the brainstem. You have 3 sensory columns sitting laterally on the outside edges and 3 motor columns sitting medially near the midline.
So suddenly, that chaotic jumble of the adult brainstem isn't chaotic at all?
Not at all. If you know the embryology, you know exactly where to look. If you are searching for a motor nucleus, you look near the midline. If you are searching for a sensory nucleus, you look laterally.
Let's test that geography by diving into a couple of specific columns, because the internal routing is just mind-bending. Let's start with the somatic sensory column.
Okay.
So this is sitting laterally. This is where that massive trigeminal nerve we talked about earlier, the one covering the whole face, sends all its raw data. It feeds into a structure called the spinal trigeminal nucleus.
The spinal trigeminal nucleus is a towering column of cells. It is essentially a direct continuation of the sensory dorsal horn of the spinal cord, and it extends upward through the entire medulla. It acts as the primary receiving station for pain and temperature information from the entire face, the nasal cavities, and the oral cavity. But what is truly remarkable is its somatotopic organization.
Meaning how the body is mapped out on the brain cells.
Exactly, how the physical topography of your face is mapped onto the neurons of this nucleus.
The text mentions the map is upside down.
It is entirely inverted. The sensory fibers innervating the forehead and the upper scalp project to the most ventral or bottom portion of the nucleus. Meanwhile, the fibers innervating the jaw and the oral cavity project to the most dorsal or top portion.
That's bizarre.
It is. And the routing of the cables before they even reach this map is a classic anatomical trap for medical students.
Because the cables loop downward before they go up. Walk us through that pathway, because it sounds impossible.
It really does. When the primary sensory axons of the trigeminal nerve enter the brainstem at the level of the pons, they don't immediately cross over to the other side and head up to the cortex.
Which is what you would expect.
Right. Instead, the fibers carrying pain and temperature physically turn downward. They form a descending bundle called the spinal trigeminal tract. They travel down the side of medulla, and they actually cross the border into the upper segments of the cervical spinal cord.
Down into the neck.
Down into the neck. They synapse onto the nucleus there, and only then do the secondary neurons cross the midline of the spinal cord, join the spinothalamic tract, and begin their ascent all the way up to the thalamus.
Wait, I really want to make sure I grasp the physical reality of this. If I burn my tongue on a hot cup of coffee, the electrical pain signal travels through my jaw, enters my brainstem, physically drops down into my neck, crosses the midline of my spinal cord, and then shoots back up through the brainstem to my cortex.
That is exactly what happens.
That is wild. And there's a massive clinical implication here for spinal cord injuries, right?
There absolutely is. If a patient suffers a traumatic injury or a lesion specifically in their upper cervical spinal cord high up in the neck, they will obviously experience deficits in their body. But because of this looping trigeminal tract, one of the presenting symptoms can actually be a profound loss of pain and temperature sensation in their face.
To a layperson, a neck injury causing facial numbness seems neurologically impossible.
Totally impossible. But once you understand the embryological routing of that specific sensory column, it makes perfect logical sense.
Okay, that covers the external somatic data. What about the internal data? We talked about the vagus nerve constantly monitoring the stretch of the stomach and the chemical makeup of the gut. How does the brainstem organize the visceral sensory data?
That information routes laterally to the visceral sensory column, specifically to a highly specialized structure called the nucleus of the solitary tract.
The solitary tract.
Yes. If you want to talk about biological engineering, the solitary tract is a masterclass in data integration. It acts as a central hub. It gathers visceral afferent fibers from cranial nerve cecum, which carries taste, cranial nerve naix, which monitors blood oxygen and pressure in the carotid artery, and the vagus nerve, which carries that massive amount of gastrointestinal, respiratory, and cardiac data.
So it's pulling taste from the tongue, oxygen levels from the neck, and fullness from the stomach.
All into one physical location. As these diverse sensory fibers enter the brainstem, they literally bundle together to form the solitary tract. They run parallel to the nucleus and synapse onto it.
Yeah.
As a result, sensory information from wildly different internal organ systems is merged to produce a single, highly integrated, unified map of the internal body's physical state.
It is the ultimate internal dashboard.
Precisely. And from there, the solitary nucleus doesn't just sit on the data. It projects this internal state map extensively. It sends fibers upward to the hypothalamus and the amygdala to influence our emotional and hormonal states, and it projects deeply into the reticular formation of the brainstem to instantly trigger autonomic reflex responses.
So we can clearly see the familial resemblance. The brainstem builds heavily on the spinal cord's basic plan, sensory plates, motor plates, longitudinal columns. But it's a heavily modified chassis. The neuroanatomy literature highlights three major architectural deviations where the brainstem dramatically departs from the spinal cord model. What is the first major upgrade?
The first major difference is internal geography regarding the long-distance cables. In the spinal cord, the long ascending sensory tracts going to the brain and the descending motor tracts going to the body run strictly on the outside perimeter of the cord.
Right, forming the white matter.
Exactly, the white matter that surrounds the butterfly-shaped gray matter core. But in the brainstem, these massive ascending sensory tracts like the medial lemniscus, which carries touch and proprioception, are incorporated deep within the interior core of the brainstem itself.
So the cables move from the outside of the tube to the inside of the tube.
Correct.
What is the second major difference?
The addition of entirely new superimposed structures that the spinal cord simply doesn't have to deal with. In the brainstem, particularly across the midbrain and the pons, you have the massive presence of the cerebellum and all of its associated feedback pathways.
The cerebellum is that dense, cauliflower-looking structure hanging off the back.
Right. And it requires immense computational pathways. Millions of cortical neurons project down to the pontine nuclei, which then send their axons across the midline and into the cerebellum to coordinate complex movements.
That's a lot of wiring.
A massive amount. These fibers, along with the descending motor tracts from the cortex, bundle together to form a massive new ventral layer on the front of the brainstem. You see this as the cerebral peduncles in the midbrain, the huge bulging base of the pons, and the pyramids in the medulla.
Wow.
This massive ventral motor porch simply doesn't exist in the spinal cord plan.
It's like taking a standard hallway and bolting a massive multi-lane highway onto the front of it.
That's exactly what it looks like structurally.
And the third deviation.
The third difference is fundamentally developmental. During embryonic development, the architecture of the spinal cord is heavily influenced by adjacent blocks of mesodermal tissue called somites.
Oh, somites.
Yes. These somites dictate the development of the vertebrae and the muscles, and they force the spinal nerves to organize into that repeating, ladder-like segmented pattern we see in adult spinal nerves. C1, C2, C3, and so on.
The segment.
Right. The hindbrain, however, does not develop under the influence of somites. Therefore, in the adult brainstem, you do not see a repeating segmented pattern of nerves. The organization is strictly longitudinal and columnar, driven by entirely different molecular gradients.
Okay, so we have fully mapped the hardware. We know where the cables go, we know how the columns sort the sensory and motor data, and we see how the dashboard integrates our internal organs. But hardware is useless without software.
True.
We need to look at how these columns actually produce behavior without needing the big, highly evolved cerebral cortex to micromanage everything. This is where the brainstem shifts from being a relay station to an independent command center.
And this brings us into the reticular formation. It's the complex web-like core of the brainstem that sits between those distinct columns and tracks. And to truly grasp what the reticular formation does, we have to look at an observation made by Charles Darwin.
Which is such a striking pivot. I mean, we are deep in the weeds of cellular neuroscience, looking at tracks and nuclei, and suddenly we're pulling from Darwin's 19th century evolutionary observations.
Yeah.
But it connects perfectly.
Darwin was incredibly prescient. In 1872, he published The Expression of the Emotions in Man and Animals. In it, he meticulously documented how mammals across vastly different species, dogs, chimpanzees, humans, share virtually identical facial expressions for core primal emotional states.
Like fear, anger, disgust.
Exactly. Surprise, too. If a dog snarls in anger, it pulls back its lips and bares its teeth using the exact same mechanical muscle activations a human uses when sneering in rage.
And Darwin's hypothesis was that because these expressions are so universal across species and even present in infants who haven't had time to learn social cues, they couldn't be learned behaviors.
Right, they couldn't be cultural.
They had to be hardwired. They had to be anatomically embedded in the lower, more ancient parts of the brain that all mammals share.
And modern cellular neuroscience completely validates his hypothesis. We now know definitively that these complex, coordinated emotional expressions are generated by specific ensembles of local circuit interneurons located entirely within the brainstem's reticular formation.
Interneuron.
Yes, we call these neuronal ensembles pattern generators.
Pattern generators. It literally sounds like a piece of software.
That is exactly what they are. They are pre-installed neural programs that produce stereotypic innate motor responses. You do not need a cerebral cortex to bare your teeth in anger or to widen your eyes and gasp in fear.
So the cortex is out of the loop.
For the initiation, yes. The brainstem receives the emotional trigger from the amygdala or hypothalamus, activates the pattern generator, and perfectly coordinates the firing of the facial nerve, the trigeminal nerve, and the respiratory center simultaneously. The software is built in.
And these pattern generators aren't just limited to facial expressions, right? They are the underlying engine for everything from simple, rapid reflexes to incredibly complex, life-sustaining behaviors.
No, absolutely.
Let's start with a simple reflex, the pupillary light reflex. If a doctor shines a pen light into your eye, your pupil instantly constricts. What is the exact mechanical pathway of that software program in the brainstem?
It is a stunningly elegant, incredibly fast loop. First, the physical light enters the eye and hits the retina, but it doesn't just activate the traditional rods and cones we use for conscious vision.
Okay, what does it hit?
There is a small subset of retinal ganglion cells that act as pure luminance detectors. They can detect light on their own because they contain a unique photopigment called melanopsin.
So wait, let's pause there. Even if someone is completely clinically blind because their rods and cones have degenerated, their eye can still detect if the sun is out.
Yes, exactly. If those melanopsin-containing ganglion cells are intact, the brain still receives ambient light data. These specialized cells send their axons down the optic tract and bypass the visual cortex entirely.
Straight to the brainstem.
Right. They project straight to a tiny relay station in the midbrain called the olivary protectal nucleus.
So the protectal nucleus is the sensor receiving the bright light alarm. What does it do with it?
The neurons in the protectal nucleus immediately send their axons bilaterally, meaning to both sides of the brain stem through a structure called the posterior commissure. And they synapse onto the motor command center for the pupil, which is the Edinger-Westphal nucleus.
The Edinger-Westphal nucleus. That's part of the oculomotor nerve complex, right?
Correct. It contains the parasympathetic preganglionic neurons. The moment they receive the signal from the protectal nucleus, they fire an action potential down the oculomotor nerve.
Bam.
Exactly. The signal hits the ciliary ganglion just behind the eye, which then commands the pupillary sphincter muscle in the iris to contract. The pupil shrinks, protecting the retina from light damage.
And that all happens in the background.
Yes, that entire complex calculation sensing light, crossing the midline, triggering a parasympathetic motor response, happens in a fraction of a second, governed entirely by the midbrain.
Okay, that is a local reflex for the eye. But what about something massive, systemic, and autonomic, like regulating blood pressure?
That's a great example.
Because every time we stand up from the couch, gravity pulls our blood down toward our legs. Why don't we immediately pass out from lack of oxygen to the brain?
Because of the baroreceptor reflex. It is a vital, non-stop survival mechanism managed by a pattern generator in the medulla. Your brain requires a massive, uninterrupted flow of oxygenated blood.
Obviously.
To ensure this, the brainstem has to constantly monitor the physical stretch of your major arteries. As blood pressure fluctuates, the arterial walls literally stretch. The vagus nerve monitors the stretch receptors in the aortic arch directly above the heart.
Okay.
Simultaneously, the glossopharyngeal nerve monitors the carotid sinus, which is the main artery in your neck feeding the brain.
So they're functioning as internal mechanical pressure gauges.
Exactly. When you stand up rapidly, blood pools in your legs, the pressure in your upper body drops, the arterial walls in your neck and chest stretch less.
Because there's less volume pushing against them.
Yeah, right. The vagus and glossopharyngeal nerves instantly detect this decrease in stretch and relay the low pressure alarm to the visceral dashboard we discussed earlier, the nucleus of the solitary tract.
The dashboard sees the pressure dropping. So how does it fix it?
The solitary tract immediately projects to a pattern generator located in the ventrolateral medulla. This network acts like an emergency response coordinator. It executes two simultaneous opposing actions.
What's the first?
First, it decreases the parasympathetic outflow to the heart via the vagus nerve. The vagus nerve normally acts as a brake on the heart rate. By lifting that brake, the heart instantly speeds up.
And the second action?
Second, it dramatically increases the sympathetic outflow down the spinal cord to the blood vessels, commanding them to constrict.
So the heart pumps faster, the pipe squeezes tighter, blood pressure spikes back up, and your brain stays perfectly oxygenated.
Exactly. All before you've even fully stood up, and all completely beneath your conscious awareness.
That is the power of a brainstem pattern generator.
It's flawless engineering.
But reflexes are one thing. I want to talk about how we coordinate behaviors that require multiple distinct actions happening in sequence. Eating is the perfect example.
Oh, eating is incredibly complex.
When you actually break down the mechanics of eating, it is a terrifyingly dangerous activity. I mean, you are taking solid matter, grinding it up, and forcefully shoving it down a muscular tube that sits mere millimeters away from your airway. A slight miscalculation means you'd choke and die.
Right, the margin of error is basically zero.
So how does the brainstem coordinate this sequence of chewing, licking, and swallowing without a hitch?
It achieves this by pooling multiple local pattern generators together and forcing them to communicate. The motor patterns are actually segregated.
Segregated how?
Well, the rhythmic alternating tongue movements for licking are organized by a network near the hypoglossal nucleus. The powerful rhythmic jaw movements for chewing are generated by circuits near the trigeminal motor nucleus.
Okay.
And the incredibly complex sequence of suck it and swallowing is managed by interneurons near the facial and ambiguous nuclei.
So you have these distinct software programs for the tongue, the jaw, and the throat. But they obviously can't operate in a vacuum.
Exactly. They are heavily interconnected. They talk to each other constantly to ensure sequence and timing. And crucially, they rely on constant real-time sensory feedback from the nucleus of the solitary tract.
The dashboard again?
Yes. The solitary tract is actively evaluating the taste, size, and texture of the bolus of food in your mouth.
Wow.
The processing power here is so robust that experimental studies show a decerebrate rat. So a rat that has had its entire cerebral cortex surgically separated from its brainstem is still capable of making sophisticated choices.
Wait, a rat without a cortex can choose what to eat?
Yes. It can evaluate a liquid placed in its mouth and, based entirely on taste and texture, execute the pattern to swallow it if it is nutritious, or reject it if it is unpalatable.
The brainstem is doing all the food critique and physical coordination entirely on its own.
Completely independently.
Which brings up the absolute opposite of swallowing. Rejecting food. Vomiting. And exploring this reflex brings us to one of the absolute strangest anatomical structures in the entire central nervous system, the area postrema.
Ah, the area postrema. It is fascinating because it intentionally breaks the cardinal rule of brain anatomy.
How so?
It is a small, specialized region located adjacent to the solitary tract, right along the floor of the fourth ventricle. Its evolutionary job is to detect toxic substances, poisons, or dangerous metabolic byproducts circulating in the blood.
But the brain is normally protected from the blood. I mean, we have the blood-brain barrier.
Right. Almost the entirety of the central nervous system is shielded by the blood-brain barrier, a tight physiological seal that prevents pathogens and large molecules in the blood from entering the brain tissue.
Yeah.
But the area postrema lacks a blood-brain barrier entirely. Instead, it contains fenestrated capillaries, blood vessels, with literal tiny windows or pores in them.
It sits there and actively tastes the raw, unfiltered blood.
It monitors the blood directly. And if the neurons in the area postrema detect high levels of a circulating toxin, say, a poison you ingested, or even certain chemotherapy drugs, they trigger an emergency response.
And where does that signal go?
They project to a specific pool of neurons in the ventrolateral medulla. This network acts as the master pattern generator for emesis, or vomiting.
And vomiting is an incredibly violent, whole-body motor event. It's not a subtle reflex.
It is massive. The pattern generator coordinates a systemic physiological reversal. It halts normal gastric digestion entirely. It triggers a reversal of intestinal peristalsis to move contents back into the stomach.
Wow.
It causes forceful, spasmodic contractions of the abdominal muscles and the diaphragm and forces the esophageal sphincter to open, violently expelling the contents of the stomach.
It is the ultimate physiological eject button, and it perfectly demonstrates how the brainstem prioritizes immediate, raw survival above all else. But if we're talking about survival, there is one pattern generator that stands above the rest, the absolute masterpiece of the brainstem: breathing.
Without question, respiration is the defining rhythm of life. From approximately the end of the first trimester of gestation until the exact moment of death, the respiratory pattern generators fire automatically, continuously, without requiring a single ounce of conscious effort.
And mapping out the architecture of breathing is incredible, because it's not just one nucleus keeping the beat, right? It's a highly networked, redundant system. Let's break down the components. It starts with a pacemaker.
Yes. The rhythm begins in a specialized cluster of neurons located in the ventrolateral medulla called the pre-Butzinger complex.
The pre-Butzinger complex.
Right. This complex acts as the central pacemaker. It generates the intrinsic rhythmic electrical bursting that sets the baseline cadence of respiration. It is the metronome.
Okay, so it sets the beat.
But a raw rhythm isn't enough to keep a complex organism alive. You need to integrate sensory feedback, and you need to execute the physical muscle contractions.
Which brings in the dorsal and ventral respiratory groups.
Correct. The dorsal respiratory group, which is located in the medulla closely associated with the solitary tract, is the primary sensory integrator.
What is it sensing?
It continuously receives feedback from stretch receptors in the lungs, essentially telling the brain when the lungs are fully inflated so you don't overinflate and pop them.
That's important.
Very. As well as input from peripheral chemoreceptors monitoring oxygen and carbon dioxide levels in the blood. It takes all that data and modulates the pacemaker's rhythm.
And then the ventral respiratory group does the physical labor.
Yes. The ventral respiratory group contains the premotor neurons that actually execute the command. They project descending axons down the spinal cord, primarily to the phrenic motor nucleus in the cervical spine.
Okay.
The phrenic nerve then fires, causing the massive sheet of the diaphragm muscle to physically contract, pulling air into the lungs.
Okay, here is where I want to push back on the neuroscience, or at least explore a massive caveat. We keep using the terms hardwired, autonomic, and stereotypic.
Right.
We are describing a system that runs completely on autopilot.
Right.
But I can choose to take a deep breath right now. I can choose to hold my breath. Or, more importantly, I can speak. Speaking requires me to completely override the metronome and release air in highly specific, modulated bursts. If this generator is an autonomic survival loop, how on earth does it allow for my voluntary cortical control?
That is the essential tension of the respiratory system. It has to serve dual, sometimes conflicting functions. It absolutely must maintain chemical homeostasis, keeping oxygen up and carbon dioxide down. But it also must act as the bellows for voluntary behaviors like vocalization, eating, and sniffing.
So how does it manage both?
This incredibly delicate flexibility is managed by a higher-level regulatory center located in the pons, known as the pontine respiratory group, or historically, the pneumotaxic center.
So if the pre-Betzinger complex is the metronome keeping the beat, the pneumotaxic center feels more like the conductor deciding how we use that beat.
That is a perfect analogy. The pontine respiratory group coordinates the transition between inspiration and expiration, and it seamlessly integrates the breathing rhythm with other complex behaviors.
Like talking.
Exactly. When you decide to speak or sing a long note, descending motor pathways from your cerebral cortex project down to the pons and essentially borrow the steering wheel.
They borrow it.
The cortex tells the pontine center to temporarily alter the rhythm, extending the exhale so you can vibrate your vocal cords. Anticipation of exercise from your limbic system can tell the center to hyperventilate before you even start running.
So the cortex can borrow the steering wheel, but it can't keep it forever.
Never forever. The brainstem is the ultimate boss. The cortex must ultimately yield to the brainstem's ironclad mandate for chemical homeostasis.
And that chemical mandate is not actually driven by a lack of oxygen, which surprises a lot of people. It is driven by carbon dioxide.
Exactly. The primary physiological driver of respiration is the clearance of carbon dioxide, which dictates the pH of your blood. If you decide to hold your breath, your cortex is actively inhibiting the inspiratory motor neurons.
Okay.
But as you hold it, the carbon dioxide produced by your cellular metabolism begins to rapidly accumulate in your blood.
The blood becomes more acidic.
Yes. And that acidity is instantly detected by highly sensitive central chemoreceptors located on the ventral surface of the medulla. As the CO2 levels rise even slightly, these chemoreceptors begin bombarding the respiratory networks with excitatory signals.
Demanding a breath.
They generate an overwhelming visceral sensation of severe air hunger, clinically known as dyspnea. It becomes psychological torture. No matter how much sheer cortical willpower you exert, the brainstem will eventually break your inhibition.
It just forces it.
It will force a massive contraction of your diaphragm to expel that CO2. It is an evolutionary marvel, a system flexible enough to let an opera singer perform an aria, but rigid enough to absolutely guarantee you stay alive.
Which is a phenomenal transition into the final piece of the puzzle for this deep dive. We have mapped the wiring, the cranial nerves, we've examined the hardware columns, we have explored the hardwired software, the pattern generators executing these survival behaviors.
Right.
But the brainstem doesn't operate in a static vacuum. A reflex that saves your life when you are sprinting from a predator might be entirely inappropriate when you are falling asleep. The brainstem has to constantly adjust the volume or the sensitivity of all these interconnected networks based on our state of arousal, our stress levels, and our environment.
And this global adjustment is handled by a completely different class of neural architecture, the chemical modulatory systems. The story of how we actually discovered the system is one of the great triumphs of modern neuroanatomy.
And it centers heavily on a researcher named Hans Kuipers in the 1970s, right?
It does.
Let's dig into that history, because Kuipers wasn't just looking at regular stained slides. He utilized a relatively new technique at the time called retrograde axonal transport. What exactly was he doing?
Well, before Kuipers, neuroanatomists struggled to map long-distance connections in the brain. They could see where a nerve started, but tracking tiny, branching axons across the entire brain was nearly impossible.
Like trying to trace one wire in a tangled server rack?
Exactly. Retrograde tracing changed that entirely. Kuipers injected an enzyme called horseradish peroxidase directly into the spinal cord.
Horseradish peroxidase.
Yes. This enzyme is taken up by the axon terminals and is physically transported backward retrogradely up the axon all the way to the cell body.
So instead of trying to follow the wire from the source, he dropped dye at the destination and waited to see which cell bodies in the brain lit up.
Precisely. And when Kuipers looked at the brain stems of these animals, he discovered something entirely unexpected. He found a massive, highly organized, but previously unrecognized neural substrate.
What did it look like?
He identified specific small groups of cells deep in the reticular formation of the brainstem that were sending axons incredibly long distances. But they weren't sending point-to-point wires. No, they were sending highly collateralized, wildly branching webs of axons down into the spinal cord, up into the cerebral cortex, into the thalamus, literally everywhere.
Wow. It was described as a parallel nervous system, right? An autonomic system for the brain itself.
Because of how diffusely it projects, yes. And what truly separates these neurons from the rest of the brain is their chemistry. These are the monoaminergic neurons.
Monoamines. We hear these terms all the time in psychology and pharmacology. Serotonin, dopamine, norepinephrine, histamine.
These are the chemical messengers. And the way these monoaminergic neurons communicate is fundamentally different from the fast, binary firing we associate with traditional neurobiology.
Let's contrast that. A traditional neuron in the motor cortex trying to move a finger uses fast synaptic transmission.
Right. It releases a neurotransmitter like glutamate directly across a microscopic synaptic cleft. It binds to an ionotropic receptor, opens an ion channel, and instantly triggers an electrical spike in the next cell.
Just on or off.
It happens in milliseconds. It is a highly specific point-to-point binary system.
But the monoamines don't do that.
Most of them do not. Instead of forming tight, targeted synapses, monoaminergic axons often feature a series of swellings called varicosities.
Varicosity.
Yes. As the electrical signal travels down the axon, these varicosities release serotonin or norepinephrine diffusely into the extracellular space. This is called volume transmission. It literally bathes wide swaths of neural tissue in the chemical.
Like a sprinkler system rather than a targeted hose.
Exactly. Furthermore, the receptors they target are mostly metabotropic, meaning they are coupled to G proteins inside the cell.
What does that mean functionally?
When serotonin binds to these receptors, it doesn't instantly fire the cell. Instead, it triggers a slow cascade of intracellular chemical reactions that alter the cell's ion channels over seconds, minutes, or even hours.
So if the fast glutamate synapses are flipping a light switch on or off, these monoaminergic systems are slowly turning a massive dimmer dial, adjusting the baseline excitability of millions of neurons simultaneously.
Exactly. They set the tone. They optimize the state of the nervous system to match the behavioral demands of the moment.
Let's ground this in reality by walking through specific, deeply consequential examples from the research. Let's go back to breathing and carbon dioxide.
All right.
There is a profound connection between a specific monoamine system in the brainstem and one of the most heartbreaking tragedies in medicine. Sudden infant death syndrome, or SIDs.
This is perhaps one of the most vital clinical insights derived from studying the brainstem. In the medulla, clustered along the midline, are the Raphi nuclei. Specifically, groups of serotonergic neurons designated as the B1 through B4 groups.
B1 through B4.
Yes. These exact neurons project heavily to the respiratory pattern generators we just talked about. And what makes them unique is that they are intrinsically chemosensitive. They are literal carbon dioxide sensors.
So they aren't just releasing serotonin, their dendrites are physically wrapping around major medullary arteries, directly sampling the blood gas?
Yes. If a baby is sleeping face down, buried in a soft mattress, they can begin rebreathing their own exhaled air.
As they do, the carbon dioxide levels in their blood begin to rise. This rise in CO2 causes a proportional drop in the pH of the blood, and the cerebrospinal fluid becomes more acidic. These B1-B4 serotonergic neurons instantly detect this acidification.
And how do they react?
They dramatically increase their firing rate, releasing a surge of serotonin.
And that serotonin flood does two critical things to save the baby's life, right?
First, it heavily stimulates the respiratory pattern generators in the medulla, forcing the infant to take faster, deeper breaths to try and blow off the CO2.
Okay, that makes sense.
Second, and crucially, it stimulates the ascending arousal networks projecting to the cortex. It forces the infant to wake up, break out of deep sleep, and physically turn their head or cry to clear their airway.
It is a chemical alarm bell. But the prevailing hypothesis for SIDs is that in some tragically vulnerable infants, this specific serotonergic alarm system is fundamentally defective.
Yes. Extensive post-mortem analysis of infants who succumbed to SIDs has revealed significant abnormalities in this exact medullary serotonergic system. What kind of abnormalities?
Pathologists find an immature morphology in the Raphi nuclei, abnormally low levels of tissue serotonin, and major deficits in serotonin receptor binding.
So the hardware is broken.
Yes. The working theory is that when these vulnerable infants experience an asphyxiating event during sleep, like being face down, the CO2 rises, but the defective serotonergic neurons fail to sound the alarm.
Oh, wow.
The pattern generators aren't stimulated to increase breathing, and the arousal network fails to wake the baby up.
The physiological implications of this are staggering.
Yeah.
Because when you realize the vulnerability lies in a microscopic chemical sensor deep in the brainstem, it entirely explains the success of the Back to Sleep campaign.
Absolutely.
Telling parents to put infants on their backs wasn't about the lungs. It was an environmental intervention designed to protect a developing brainstem from ever having to rely on a potentially faulty CO2 alarm. That completely changes how I view infant sleep hygiene.
It is a prime example of how mapping an obscure brainstem pathway can translate into a massive public health victory.
Let's shift to another massive modulatory function: pain. The text details how these monoamine systems don't just amplify signals, they can profoundly suppress them.
Yes, through what we call descending anti-nociceptive pathways. We typically think of pain as a purely bottom-up phenomenon. You touch a hot stove, the signal goes up the spinal cord to the brain, and you feel pain.
Right, a straight line.
But the brainstem has the ability to actively shut that signal down before it ever reaches consciousness. Both noradrenergic neurons from the pons and serotonergic neurons from the Rafu nuclei send dense axonal projections all the way down to the dorsal horn of the spinal cord.
Which is the exact spot where incoming pain fibers synapse.
Correct.
And what do they do when they get there?
They release serotonin and norepinephrine to actively inhibit the transmission of noxious stimuli. From an evolutionary standpoint, this is essential.
How so?
If an animal is fighting for its life or fleeing a predator, feeling the crippling, conscious pain of a torn muscle is maladaptive. It will get the animal killed. The brainstem recognizes the extreme stress state, floods the spinal cord with monoamines, blunts the pain signal at the source, and allows the animal to keep running.
It's an endogenous painkiller system. And pharmacologically, modern medicine has figured out how to hijack this exact mechanism to treat severe migraines.
Exactly. Triptan drugs, like sumatriptan, revolutionize the treatment of migraines.
How do they work?
Migraines involve excruciating pain signals traveling via the trigeminal nerve from the inflamed meninges surrounding the brain. Triptans are highly specific agonists for the 5-HT1B and 1D serotonin receptors.
They act like serotonin.
Right. By flooding the system with these serotonin mimics, the drugs chemically replicate the brainstem's descending inhibitory signal, effectively blocking the pain afferents from transmitting the migraine signal to the cortex.
We are just chemically pressing the brainstem's mute button. Incredible. Now let's talk about the motor system. Everyone casually associates dopamine with movement because of the prevalence of Parkinson's disease. But the modulatory mechanics are incredibly specific.
Dopamine is critical for movement initiation. Massive dopaminergic projections originate from a structure in the midbrain called the substantia nigra. They ascend heavily into the striatum of the basal ganglia. Dopamine acts as the essential lubricant for the motor circuits. It facilitates the selection and initiation of voluntary movements. When those specific dopamine-producing cells degenerate and die, you see the devastating clinical hallmarks of Parkinson's: severe rigidity, resting tremors, and akinesia.
Akinesia meaning an inability to initiate movement.
Right.
But the textbook also details norepinephrine role in movement. And this relates to a phenomenon almost everyone has experienced, the stress tremor.
Oh, yes.
When you are terrified or incredibly anxious, your hands physically shake. Why does that happen?
Norepinephrine acts as an excitatory modulator for the motor system. Noradrenergic inputs descending from the locus ceruleus in the pons synapse onto motor neurons in the spinal cord, and their job is to facilitate excitatory inputs, particularly for repetitive or sustained muscle contractions.
Okay, so they amp up the muscle tension.
But when you are placed under severe psychological stress, say public speaking or a highly competitive event, your locus ceruleus goes into complete overdrive. You experience a massive systemic surge of beta-adrenergic activation.
You flood the engine with too much norepinephrine.
Exactly. This exaggerated chemical facilitation overstimulates the motor neurons. The muscles receive way too much excitatory noise, producing a physical, high-frequency resting tremor. Your hands shake not because you are weak, but because your motor neurons are hyperexcitable.
And this physiological reality is exactly why classical musicians and public speakers often use a specific chemical hack. They take beta blockers before a major performance.
Yes, drugs like propranolol physically block those beta-adrenergic receptors on the motor neurons.
So the brainstem is still screaming in panic.
Right, releasing massive amounts of norepinephrine due to the stress. But the beta blockers prevent the chemical from binding to the muscles. It artificially mutes the brainstem's stress response at the muscle level, keeping the musician's hands perfectly steady to play a delicate violin concerto.
It is entirely mechanical. Speaking of the locus coeruleus and norepinephrine, let's dive deeper into how it affects our psychology.
Mm.
Because this tiny cluster of cells fundamentally dictates our attention and our focus. The research outlines how the locus coeruleus operates in two distinct firing modes. Walk us through that.
The locus coeruleus is a tiny nucleus, but its axons reach virtually every part of the cerebral cortex. It dictates arousal and attention. Experimental recordings in alert monkeys demonstrate it has two distinct behavioral states, a phasic mode and a tonic mode.
What's the phasic mode?
In the phasic mode, the baseline firing rate of the neurons is very low and quiet. But the moment the monkey is presented with a specific, rewarding task, like a light that cues them to press a lever for juice, the locus coeruleus neurons fire in massive, sharp, synchronized bursts just prior to the behavioral response.
So the phasic burst represents locked-in, highly focused attention on the task at hand. The brain saying, pay attention to this exactly right now.
Exactly. It optimizes cortical processing for that specific goal.
Right.
But what happens when the environment changes?
Right.
Say the researchers stop providing the juice. The task is no longer rewarding. The monkey becomes frustrated and distracted. At this point, the locus coeruleus drastically shifts its firing pattern. It enters the tonic mode.
What does that look like?
The overall baseline firing rate becomes continuously highly elevated, but it stops producing those sharp bursts to the task cues.
It stops focusing and starts scanning.
Precisely. The elevated tonic firing promotes generalized arousal and distractibility. It facilitates scanning the environment for new stimuli. It is the brainstem chemically signaling the cortex. The current strategy is failing. Disengage your focus and find a new behavioral goal.
And the calculation of what constitutes a rewarding goal is driven by dopamine, right?
Mm-hmm.
We mentioned dopamine for movement, but its role in motivation is equally profound, specifically through reward prediction errors.
Oh, absolutely. Dopaminergic neurons originating in the ventral tegmental area project heavily to the prefrontal cortex and the limbic system. For a long time, we thought dopamine was just the pleasure chemical it fired when you got a reward.
That's the pop science version.
Right. But advanced recordings show something far more sophisticated. Dopamine neurons fire primarily when a reward is unexpected. If an animal unexpectedly finds food, dopamine spikes. It is a learning signal.
Hey, whatever you just did, remember it.
Exactly. But once the animal learns that a specific cue, like a ringing bell, predicts the food, the dopamine spike actually moves. It stops firing when the food arrives and instead fires precisely when the bell rings. The dopamine is tagging the cue with incentive value. It teaches the brain what to desire, driving the motivation to seek it out.
And if you combine all these monoamines, the serotonin, the dopamine, the norepinephrine, the histamine, they operate as a unified symphony to control the most fundamental cycle of human existence.
Yeah.
Consciousness and the sleep-wake cycle.
Together, they form the ascending arousal network. They project massively from the brainstem through the thalamus, bathing the entire cortex. And across the daily circadian cycle, they share a remarkably consistent firing profile.
How does it fluctuate?
When you are fully awake and alert, these monoaminergic neurons fire at their highest rates, keeping the cortex desynchronized and receptive to sensory input. As you get drowsy and enter slow-wave non-REM sleep, their firing rates progressively slow down.
And during REM sleep, when we are deeply dreaming.
The serotonergic and noradrenergic neurons almost completely cease firing. They fall utterly silent. The brainstem chemically isolates the cortex, allowing the intense, internally generated cortical activity of dreams to occur without external sensory interruption or physical motor acting out. They literally set the tempo of human consciousness.
Which brings up a phenomenal pharmacological paradox highlighted in the clinical literature regarding Parkinson's disease. We discussed how Parkinson's involves the death of dopamine neurons in the substantia nigra.
Right.
So the standard treatment is to give patients drugs, dopamine agonists, that artificially stimulate dopamine receptors to restore their ability to move.
Yes, drugs that specifically target the D2 subtype of dopamine receptors.
But a major, highly frustrating side effect of these movement-restoring drugs is severe daytime sleepiness. Patients often suffer from sudden, overwhelming sleep attacks. On the surface, that makes absolutely no sense. Why would a drug designed to restore motor function by mimicking an activating chemical force you to fall asleep?
It is a perfect illustration of the unintended consequences of chemical modulation. The brain is an incredibly complex web. The therapeutic goal of the drug is to act on postsynaptic D2 receptors in the striatum to facilitate motor pathways. And it does that very well. The patient's rigidity decreases.
What's the catch?
The catch is where else those D2 receptors are located. Those same D2 receptors also exist as presynaptic autoreceptors on the remaining healthy dopaminergic neurons in the ventral tegmental area.
The neurons responsible for arousal and motivation.
Exactly. Autoreceptors function as a negative feedback loop. They tell the neuron how much transmitter is in the synapse. When the patient takes the synthetic dopamine drug, it binds to these autoreceptors.
And the autoreceptor falsely signals the cell, hey, we are flooded with dopamine out here. Shut down production immediately.
Precisely. The drug heavily inhibits the endogenous, natural release of dopamine from the arousal networks. The motor disorder improves because the striatum is artificially stimulated, but the arousal system is inadvertently chemically suppressed. The patient can move their arms again, but they fall asleep in their chair because the brainstem's wakefulness circuit has been tricked into shutting down.
Biology is just a web. You pluck one string to fix a motor tremor, and the vibration accidentally knocks out the entire sleep-wake cycle.
It demands an immense respect for the interconnected architecture of the central nervous system.
It really does. We have covered an astonishing amount of physiological ground today. We started by mapping the high-speed fiber optic cables of the cranial nerves, stretching from the delicate muscles of the eye deep into the chemical receptors of the gut. We rewound time to watch the embryonic neural tube physically splay open, sorting the sensory and motor columns into a logical, accessible dashboard for the internal body. We dissected the hardwired pattern generators, proving that the brainstem possesses the independent software to coordinate everything from the pupillar reflex to the violent expulsion of toxins and the exquisite lifelong rhythm of human breathing. And finally, we explored the diffuse, slow-acting monoamine networks, the chemical dimmers that dynamically dial our pain, our attention, and our very consciousness up and down.
It requires a profound shift in perspective. If you take away anything from this deep dive, let it be the abandonment of the lizard brain myth.
It's so outdated.
It is. The brainstem is not an evolutionary leftover. It is not a primitive relay station. It is a highly conserved, exquisitely engineered control center. It maps the physical reality of our body. It ruthlessly defends our blood chemistry and our breath. It hardwires the universal expressions of our emotions, and it bathes the entire cerebral cortex in the precise modulatory chemicals required to maintain human consciousness.
And that leads to a deeply provocative final thought I want to leave you with, based on everything we've unpacked today from Chapter 40. We spend so much of our time in philosophy and cognitive science obsessing over the enormous cerebral cortex.
Yes.
We believe the cortex to be the undisputed seat of our true self, the engine of our free will, and the source of our high-level cognition. But as Charles Darwin astutely noted with our facial expressions, and as the monoamine arousal and reward networks unequivocally prove, our most complex psychological states are fiercely focused attention. Our motivation to seek rewards, our intensely felt emotional responses are entirely reliant on and generated by these ancient stereotypic patterns firing deep in the brainstem.
It raises a fundamental, almost unsettling question about the nature of the mind. Is human consciousness actually the top-down, cortically driven phenomenon we naturally assume it is? Or is the cortex secondary? Are our complex, conscious thoughts merely the highly decorated, post-hoc echoes of our brainstem's ancient, hardwired survival mechanisms?
Are we just highly articulate narrators rationalizing the chemical dictates of our brainstem? It is something to seriously ponder as we continue this series into higher cognition. Thank you for joining us on this deep dive. We hope we've completely shattered and rebuilt how you view the architecture of your own mind. So the next time you hold your breath to swim underwater, or feel a rush of adrenaline before a big speech, or simply smile genuinely at a joke from a friend, take a moment to thank the brilliant, tireless, microscopic machinery operating continuously at the base of your skull.